Carbohydrates are essential for life. They are our primary source of energy. Our bodies break down carbohydrates into glucose. Glucose is a simple sugar that our cells can use to make ATP. ATP is like the energy currency of our cells. Without carbohydrates, our bodies wouldn't function properly. Carbohydrates are important for physical activity too. They provide the fuel our muscles need to work. From running a marathon to walking the dog, carbohydrates give us the energy to move. Our brains also rely heavily on carbohydrates. The brain requires a constant supply of glucose to function correctly. Without enough glucose, we can experience fatigue, dizziness, and difficulty concentrating. Carbohydrates play a vital role in maintaining overall health. They are an essential part of a balanced diet and provide the body with the energy it needs to thrive. Glycolysis is the process of breaking down glucose. It's a fundamental metabolic pathway that occurs in the cytoplasm of almost all living cells. The process doesn't even require oxygen. This pathway is essential for life because it provides cells with energy. Glycolysis starts with one molecule of glucose. Think of glucose as a 6-carbon sugar molecule. Through a series of 10 enzyme-catalyzed reactions, glucose is broken down into 2 molecules of pyruvate. Pyruvate is a 3-carbon molecule. During glycolysis, 2 molecules of ATP are consumed in the initial steps. However, the pathway generates 4 molecules of ATP through substrate-level phosphorylation. This results in a net gain of 2 ATP molecules. In addition to ATP, glycolysis also produces 2 molecules of NADH. NADH is a coenzyme that carries electrons. These electrons are important for generating ATP in the later stages of cellular respiration. To be utilized by our cells, glucose needs to get inside. That's where glucose transporters, or GLUTs, come in. These proteins are embedded in cell membranes and act as gatekeepers for glucose. There are different types of GLUTs, each with a specific role and location in the body. 4 key GLUTs are GLUT1, GLUT2, GLUT3, and GLUT4. Some of these transporters rely on insulin, a hormone that regulates blood sugar levels. GLUT1 is found in most cells and ensures a constant glucose supply. GLUT2, present in the liver, pancreas, and intestines, helps regulate blood glucose levels. GLUT3, found in neurons, provides the brain with glucose. Finally, GLUT4, present in muscle and fat cells, is insulin-dependent, meaning it requires insulin to transport glucose efficiently. Section 4, hexokinase versus glucokinase, first step regulators. The first step of glycolysis involves adding a phosphate group to glucose. This is called phosphorylation. 2 enzymes, hexokinase and glucokinase, can do this job, but they differ in their roles and locations. Hexokinase is found in most tissues and has a high affinity for glucose. It's inhibited by its product, glucose 6-phosphate, preventing excessive glucose phosphorylation when glucose levels are adequate. Glucokinase, primarily found in the liver and pancreas, has a lower affinity for glucose but a higher capacity. It's not inhibited by glucose 6-phosphate, allowing the liver to uptake and store glucose efficiently, even when glucose levels are high. Section 5, irreversible steps in glycolysis, control points. Glycolysis has 10 steps, but only 3 are irreversible. These irreversible steps are control points, meaning they can be regulated to speed up or slow down the entire pathway. The first irreversible step is catalyzed by hexokinase or glucokinase, as discussed earlier. The second irreversible step is catalyzed by phosphofructokinase 1, which adds a second phosphate group to fructose 6-phosphate. Phosphofructokinase 1 is the most regulated enzyme in glycolysis and is considered the committed step. The third irreversible step is catalyzed by pyruvate kinase, which converts phosphenolpyruvate to pyruvate. Pyruvate kinase is regulated by allosteric effectors and phosphorylation, ensuring the pathway operates at the appropriate rate for the cell's energy needs. Section 6, substrate-level phosphorylation, direct ATP synthesis. ATP is the energy currency of cells. In glycolysis, ATP can be generated through substrate-level phosphorylation. This process involves directly transferring a phosphate group from a high-energy molecule to ADP, forming ATP. 2 steps in glycolysis use substrate-level phosphorylation to make ATP. The first is the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, catalyzed by phosphoglycerate kinase. The second occurs during the conversion of phosphenylpyruvate to pyruvate, catalyzed by pyruvate kinase. Substrate-level phosphorylation is a crucial mechanism for ATP production in glycolysis. It provides a rapid way to generate ATP when oxygen is limited, such as during intense exercise.